The overall goal of this procedure is to describe how to perform whole cell patch clamp recordings in freshly dissected brain slices.
This method can help answer key questions in the field of neuroscience, such as how experimental controlled manipulations alter the activity of specific neurons in specific brain regions. The main advantage of this technique is that it provides the mean to identify in ex vivo preparation long-lasting changes in neuronal functions that have developed in intact, awake animals.
Though this method can provide insight into neuronal functions in brain slices, it can also be applied to other systems such as cells in culture and neurons in vivo.
Demonstrating the procedure will be Francisco Garcia-Oscos, a student from my lab.
To begin this procedure, using a plastic trimmed tip transfer pipette, gently draw up one brain slice from the recovery chamber. Place the transfer pipette in the recording chamber and gently squeeze the slice out of the pipette onto the cover slip lining at the bottom of the chamber. Next, using the microscope 4x objective, position the slice so the desired area is placed exactly in the center of the recording chamber. After the desired position has been achieved, secure the brain slice position with a slice hold down, also known as a harp. After that, switch to the 40x objective and lower the lens gently until it contacts the ACSF in the chamber. Then, use the fine adjustment wheel to bring the tissue into focus. When the focus is at tissue level, observe the cells in the targeted region. Now,